Good evening, one and all. I welcome you all to Ganesh IAS Academy. In today's session, we'll be seeing science and technology based current affairs from the date June 18th to June 25th, 2023. Okay, so let us discuss the news articles one by one. The first news that we are going to see today is paper based supercapacitors from seaweed. So, what is the news? Why is this paper-based supercapacitor is in the news? That we have to understand first. Scientists at Gujarat Energy Research and Management Institute, that is GERMI, have achieved a breakthrough in energy storage technology. So they have invented a technology, a new technology, energy storage technology, with the development of what? Paper-based supercapacitor. So from this news, what all do we have to focus for our exam is, we must know what is paper-based technology. Other than that, from what it is derived? It is derived from seaweed. So we have to focus on what is seaweed and its applications and its types and etc. Okay, let us see this. This cutting edge supercapacitor is derived from seaweed and it boasts remarkable attributes. It has remarkable features like what? Lightweight, biodegradable. This is important. And it is capable of fully charging a device within mere 10 seconds. Just in 10 seconds, it will charge the device completely. Okay, it has that much of capacity. Okay, this supercapacitors. And then this technology is versatile and eco-friendly. Okay, anything is biodegradable, that becomes eco-friendly or environment friendly. Okay, and it is also versatile because it can charge very fastly. Okay, and thus it is an attractive option for both manufacturers and we consumers. Okay, so all these points are important. Next, we'll understand some other features of supercapacitors of this particular supercapacitor, which has been recently developed by GERMI. Okay, it is thinnest and most lightweight of its kind. Okay, there are many supercapacitors, and of these, this supercapacitor is the thinnest and lightweight of all its kind. Okay, next is by leveraging cellulose nanofibers. This is important. From seaweeds, what are they going to get? They are going to get cellulose. Okay, and in cellulose they are going to take nanofibers of cellulose. Okay, so cellulose nanofibers, which is derived from seaweed, is going to be used to create what? Anodic paper capacitors. Okay, we'll understand what is paper capacitor in a short while. And it has another features, important features like tensile strength, performance and cost effectiveness. It is also cost friendly. Next is its applications, where all we can find its application, that is supercapacitors application. Obviously in electronics, because it can store energy. Okay, and then memory backup systems, airbags, heavy machineries and electrical vehicles, industries seeking high performance energy storage solutions. Okay, so whichever industry which is focusing on energy storage solutions, then they can use such supercapacitors. Now we'll understand what is supercapacitor. What is a supercapacitor? A supercapacitor is an electrochemical charge storage device. So all the words are important. It is electrochemical. Okay, they are going to use electrolytes, electrodes and other things in this. Okay, so it is electrochemical charge storage device, and they are also called as ultra capacitors. Okay, supercapacitors are also called as ultra capacitors. Next, it has significant advantages over the conventional one, like high power density, long durability, ultra fast charging. Already we saw this point, ultra fast charging. Other than that, it has high power density and also long durability. It can be used for a long period of time. And then what are the main components of any supercapacitor? It will be having electrodes. Okay, positive and negative, cathode and anode. And then it will have current collectors at this end. And then there will be electrolyte. This green portion is the electrolyte. Okay, and then in between the electrolyte, the two electrodes, there will be, in between the two electrodes, there will be a separator, electrolyte separator. Okay, and then we have charged electrolytes, ions, which are getting accumulated at one point. So what will happen in this supercapacitor is, when an external circuit is connected, then there will be accumulation of negative charge, because this is electrode, right? Electrode cathode side is negatively charged, so negative charge will be accumulated here. So these negative charges will be attracting positive ions from the electrolyte. Okay, so positive ions will be attracted, cations will be attracted. Next, at this side you have anode, positive charge, it will attract anions, negative ions. Okay, so all these points are very important. So when there is a charge accumulation at this layer is called as double charge layer. Double charge layer. When you get a complete double charge layer, then it means the battery is fully charged. I mean the capacitor is fully charged. Then we can make use of it to power something like fan, light or anything else. Okay, when we are going for discharge of ions, okay, then these charges will be discharged, and these charges, positive charges, will be discharged here, and the negative charges will be discharged. When the charges are discharged, at those times these ions will not be accumulated here because they are discharged. There is no more attraction at one end. Okay, so they will be inside this electrolyte. So this is how while charging they will come to the periphery, and then when it is not charging and when the charges are being utilized, then at those times these ions will be revolving inside. Okay, it will be contained inside this electrolyte. So this is how while charging and while the discharge is taking place, this capacitor works. Okay, so you must have to understand the basics of what is a supercapacitor. Now we have to get into the details of from where this is derived from. Okay, where from seaweed cellulose only we are going to make this new supercapacitor. What are seaweeds? Seaweeds are macroalgae which are attached to rocks or other substrata. Okay, in which areas? Coastal areas. In coastal areas you will be having rocks and even other substratum where the macroalgae will get attached. Those macroalgae are called as seaweeds, and they are classified based on their pigmentation as chlorophyta, rhodophyta and phaeophyta. Okay, what is chlorophyta? If they are based on pigmentation only we are classifying, right? So if it is green color, then that becomes chlorophyta. If it is red color, then it becomes rhodophyta, and if it is blue color, then it is phaeophyta. Okay, here you can see many examples. This is red algae, so it is rhodophyta. Kelp is actually brown algae. Okay, so it is phaeophyta. Likewise, you have green sea lettuce, so it is chlorophyta. Okay, and this supercapacitor we are going to produce using this chlorophyta. Okay, so it becomes important. Next, among these three, that is chlorophyta, rhodophyta and phaeophyta, this chlorophyta holds more potential components like carbohydrates, lipids, proteins and other bioactive components, bioactive agents it has. Okay, so chlorophyta becomes important for many applications. So what is seaweed, where it is found, what are its types, all these are important with respect to your exam. And what is the significance of seaweed, that we have to understand, and what are its important features. The first thing is it has nutritional value. Okay, it is rich in nutrients like vitamins, minerals and dietary fibers. It has many nutrients. Next is it is also having certain medicinal properties like anti-inflammatory, antimicrobial agents are present in seaweeds. Other than that, it also has certain cancer fighting agents. So all these points are important. Next is bioindicator. It is also a bioindicator. What is bioindicator? That we have to understand. Say for example, if there is discharge of wastewater into ocean from various sources like agricultural source, industrial source or aquacultural source, or even households may be a source for wastewater discharge. When there is a lot of wastewater discharge, then the nutrients in that wastewater will get accumulated at one point where it is discharged. So at those points, the nutrient richness will cause something called as algal bloom. Okay, it will cause algal blooming. Algal blooming is not a good thing for any water body, because what will happen is if there is a water body This is surface of water, and if there is algal bloom, then those algae will cover the entire stretch of the top layer of the water, so that what will happen is the sunlight will not penetrate into the deeps of water. Okay, it will not get into the deeps of water. Thus the marine life which is living inside at deeper depths, they will not get sufficient sunlight, and they will not be making use of the oxygen that is produced with the help of other phytoplanktons which is present in the topmost layer. So such problems will happen when there is algal bloom. Okay, so why are we seeing this point, bioindicator? Which is a bioindicator? The seaweed is a bioindicator because these seaweeds will absorb excess nutrients. Okay, even though if you are going to send some wastewater which has very high amount of nutrients, they will get absorbed by these seaweeds, so that it will not lead to formation of algal blooming. Okay, so algal blooming can be avoided, safely avoided, when we go for seaweed cultivation. Okay, so this is yet another important point. Next is oxygen production. Seaweed also leads to oxygen production because they are photosynthetic organisms. Okay, in the process of photosynthesis, what is consumed and what is released? Carbon dioxide is consumed and then oxygen is released, right? So it produces oxygen during the photosynthesis process, and that oxygen is used for sustaining respiration and survival of marine life. Thus it also indirectly or directly helps the marine life. Next is cellulose content. This cellulose content is what we are going to use. Okay, so green seaweed, that is chlorophyta part. Okay, the first type. Okay, in that there is something called as cellulose. Okay, a particular cellulose which is used for this purpose, and these green weeds are extensively found in India. At which place? Porbandar coast of Gujarat. Now it is being cultivated there. Okay, but very small amount. With that only they have did this research. Okay, so this cellulose is found to be the most suitable biopolymer material which is used in the manufacturing of this paper-based superconductors. Super capacitors. I'm sorry. Paper-based electrode materials are made using these cellulose materials, and also other storage applications it finds. Okay, next is cellulose itself is an insulating material. This point is important. Okay, cellulose is an insulating material, but in supercapacitors we need conducting material. So what we have to do is, these insulating material, that is cellulose, has to be coated with a conductive material, then only it can be used for the making of such supercapacitors. Okay, that distinction you must know. Cellulose is an insulating material, but it is used in supercapacitors. Okay, statements like these may be asked in your prelims because such statements may be confusing. Okay, if it is a superconductor, then it has to be a conductive material, but then actually it is insulating material. It is coated with a conductive material and then it is made into, it is put into application. Okay, so such statements become very important. Next is seaweed cultivation in India and also all over the world. All over the world, if you see, then China produces approximately 57%, which is the largest. Okay, so China holds the largest share of seaweeds production. Okay, cultivation, that is 57%, and then comes Indonesia with 28%, and it is followed by South Korea. But what is India's position? It is mere 0.01 to 0.02%, which is very less. But then in a recent estimate, okay, it is said that even if we go for approximately 10 million hectares or 5% of India's exclusive economic zone, that is the coastal zone, if those area is dedicated for seaweeds cultivation, then we can find many benefits. What are the benefits that we will be having? It provides employment to approximately 50 million people, okay, which is a great thing. Next is, it contributes to national GDP. When people get enough money, then obviously it will contribute to national GDP, and we can also go for import and export. Okay, next is, it leads to ocean productivity because it produces oxygen, which in turn will improve the marine quality, the quality of life in the marine organisms. So it increases ocean productivity. Other than that, it abates algal bloom, as we saw already. Next is it can be used for sequestration of carbon dioxide because they are photosynthetic. Okay, in photosynthesis process, carbon dioxide is absorbed. So they will be useful in sequestering carbons. Next is carbon dioxide. Next is could produce bioethanol. This point is also important. Okay, seaweeds are also used for production of bioethanol, and if we go for 10 million hectares, then we can produce up to 6.6 billion liters of bioethanol. Okay, so all these points are important. Even if a question is asked based on seaweeds in your mains examination, then you can state all these points and explain it briefly. Okay, so these points are important for your exams. Now we will go to the next article for the day. Okay, the next news article, that is eco-friendly lab-grown diamond. Okay, so what we are going to see, lab-grown diamond. Why is it in the news? Recently, our Prime Minister, Mr. Narendra Modi, has visited United States of America. Okay, as for a state visit. Okay, they have invited him, and in that, when he met the First Lady of United States, that is Jill Biden, then he presented her with this eco-friendly lab-grown diamond as a gift. Okay, so what is this eco-friendly lab-grown diamond? How is it produced? That is what is important for our exam with respect to science and technology topic. Okay, what is lab-grown diamond? They are synthetic diamonds produced in laboratory, and this point is important. They possess the same composition and physical properties as the original diamond. Okay, so the naturally occurring diamond will be having certain chemical properties and physical properties. These lab grown diamonds will also be having the same kind of properties because they are grown in such environment. Okay, so what is the method that we are going to use for production? They are manufactured through high pressure, high temperature methods, or there is yet another method called chemical vapor deposition. Okay, which also involves high temperature and very low pressure. Okay, vacuum pressure. Vacuum condition is used for chemical vapor deposition. So we have to understand what this chemical vapor deposition is, because that is the most widely used method for production of lab grown diamonds. Chemical vapor deposition, what does it do? So this is the experimental setup actually. Okay, there will be a substrate, and over it there will be diamond seed. Diamond seed here is the original diamond, the naturally occurring diamond. They will be put into the sealed chamber. This is the sealed chamber, and in that the diamond seed will be placed over this substrate. Okay, that is called as the substrate, and there will be carbon rich gas inserted, filled within this chamber. Okay, there is carbon rich gas, that is hydrocarbon gas is present, and heavy heat is supplied, that is 80 degrees Celsius up to 800 degrees Celsius up to 800 degrees Celsius. The temperature is given. Okay, and under these conditions, these gases will be there, right? These carbon rich gases, they will get accumulated over this seed. Okay, atom by atom they will get accumulated, and then they will form this lab grown diamond. Okay, diamond are carbon atoms, right? It is an allotrope of carbon, so they will get accumulated atom by atom. Those carbon atoms will get accumulated, okay, one by one. Okay, so a plasma will be formed when you supply so much of heat to that hydrocarbon gas. Okay, so all these points are important, and they find various applications. Okay, diamond has a very unique property that it is hardest material. Okay, so for that property it finds various applications in industrial tools, machineries. Okay, and also they are used as heat spreaders in electronics. Okay, so this topic is also important. So what is the method that is used? That is important for prelims point of view. Chemical vapor deposition is used for lab grown diamonds. Okay, carbon rich gas, what is the temperature range? All these are important. Next, the next topic that is there is leptospirosis. So what is leptospirosis? It is actually a disease. What kind of disease? It is a bacterial disease. It is a fatal bacterial disease. So when you study a topic which is based on disease, then you must have to know what is the cause of the disease, what are the symptoms, what is the treatment for it, and all those things, where it is found, okay, the region where it is found, all these are important. Okay, this is how we have to study. And if there are any scientific names for those particular viruses or bacteria, then that also becomes important. Okay, so what is this leptospirosis? It is a fatal bacterial disease. Okay, why it is in the news now? Because we are in monsoon season now. Okay, we are facing southwest monsoon in India. These leptospirosis disease will get spread during this monsoon season only. So it is more prevalent during monsoon months. So that is why it is becoming a major concern now. Okay, and who are getting affected by it? It is an occupational hazard for those who are working in agricultural settings and also those who are working in sanitary services. Okay, so for these two occupational people, this is becoming a major problem. That is because of this leptospirosis. Why? Leptospirosis is caused by this bacteria, that is Leptospira interrogans. Leptospira interrogans. This becomes important. It is primarily found in the urine of the infected animals. Okay, so the urine of infected animals may get contaminated at the places where agriculture works are involved and also sanitary works are involved. So those are the people who are getting affected the most. Okay, next, wild and domestic animals, both are the carriers of this leptospirosis. Okay, Leptospira interrogans. Specifically, rodents, cattle, pigs, and dogs. These are the animals which are the carriers. They serve as the carriers for Leptospira interrogans, the bacteria. What are the symptoms of this particular disease? Common symptoms like sudden fever, chills, headache, and sometimes there won't be any symptoms at all. And at extreme cases, severe cases, then it will also lead to organ dysfunctions like organs like liver, kidneys, lungs, and the brain will get affected by this leptospirosis. And how is it transmitted from one person to another? The transmission cycle begins with the infected animal shedding leptospira in their urine. Okay, so through the animal's urine only it is getting spread. Okay, when we come in direct contact with the urine or indirectly, if the urine is contaminated in the soil or in the water, and if we happen to come in direct contact with such environments like soil and water, then we may get infected by this particular bacteria. Okay, and people with cuts and abrasion are those who are at high risk of contracting this disease. Okay, so all points are important. Next is how we can prevent this. Okay, they have suggested an approach called One Health approach. So what is this One Health approach? It has been in news very recently. Like very often we can find this approach called One Health approach for addressing any health issues or environmental issues. So what is this One Health is? It considers the interconnectedness of what things: environmental health, animal health, and human health. Okay, interconnectedness of environmental health, human health, and animal health is called as One Health. Okay, that is One Health approach. So even if a question is asked in your mains based on One Health, then you can write such, you can draw such pictures and explain it. Okay, so what they are saying is you have to consider the problem because animals are involved here, animals are impacting humans here, and when humans are impacted, the environment will obviously be impacted. So that is how the holistically we have to address this problem. Okay, so we are going for One Health approach. Other than that, there is also a misconception about this leptospirosis. What is the misconception? That is only rodents, that is rats, are responsible for leptospirosis is what people have considered all these days. But then no, even certain wild animals and even other domestic animals like cattle, dogs are responsible for spread of leptospirosis. So that point is also important. So it has multiple animal reservoir host. Okay, not only rats, also other animals. So if there is a question that so if there is a statement in your prelims that only rats are responsible for spread of leptospirosis, then that statement becomes wrong. Okay, so these are important for your exam. Next, the next topic that we are going to see today is indigenous mRNA based Omicron specific booster vaccine. Okay, recently Department of Biotechnology of India. Okay, Department of Biotechnology has approved this Omicron specific mRNA based booster vaccine. What is its name? It is Gemcovac. Gemcovac OM. Okay, that is the name of this particular vaccine. Okay, and it is considered to be a big milestone in containing COVID-19 spread. Okay, and who has developed this? It has been developed by Genova Biopharmaceuticals India Limited. Okay, so Genova Biopharmaceuticals Limited using indigenous platform technology. So they have used indigenous technology. Okay. Next, it is also supported by, this development, this innovation was supported by Mission COVID Suraksha initiative, which is implemented by this BIRAC. What is BIRAC? Biotechnology Industry Research Assistant Council. Okay, they are conducting an initiative called Mission COVID Suraksha. Okay, and they are supporting this particular agency for the development of this vaccine. Okay, Gemcovac, which is for Omicron variant. Next is, this vaccine has got emergency use authorization. Okay, who's the which is the body which will be giving such authorization? The body is Drugs Controller General of India. Okay, Drugs Controller General of India is the one who authorizes emergency use authorization. Okay, this vaccine has got emergency use authorization. That point is important. Apart from that, there is yet another important feature for this particular vaccine, that is it has thermostability. What is thermostability? Earlier, when we have taken the first dose of vaccine, say for example Covishield or Covaxin, then you might have observed that they are stored in cold boxes. Okay, cold storage boxes they are stored. But here this Gemcovac does not need such cold storage facilities or cold storage infrastructure. Okay, so that it can be deployed to people very easily. Okay, it can be taken to people very easily. Such infrastructure barriers are not here with respect to this particular vaccine, which is a very good thing. Okay, so that is why it becomes important. So Gemcovac, who has developed it, and it has got emergency use authorization by which body? All this is important. Next is, it is a mRNA vaccine. Okay, we saw it is an mRNA vaccine. So this point becomes important for prelims. What is mRNA vaccine? What is RNA? And in RNA there are types. Among those types, what is mRNA? What is its specific function? That we have to understand. Okay, so let us get into the background of what is RNA. Okay. RNA, what is RNA? It is ribonucleic acid. It is a molecule with a chain of nucleotide. So what is a nucleotide? Nucleotide contains a nitrogenous base, a ribose sugar and a phosphate group. Together this is called as a nucleotide chain. Okay, and just like DNA, RNA is also very vital and important for any living cells. Okay, next is, RNA is formed from DNA. Okay, DNA will be there inside the nucleus, and from DNA only this RNA is formed with the process called, through a process called transcription. Okay, the process is transcription. So when you take when you produce RNA from DNA, then that is transcription. When the reverse happens, that is when we are going to produce DNA from RNA, then that becomes reverse transcription. Okay, so all these terms are very important, and for that process it uses a particular enzyme called RNA polymerase. Okay, next is, what is the structure of a nucleotide? This is a typical structure of a nucleotide. It will be having a nitrogenous base. Okay, and then it will be having a pentose sugar group. Okay, in case of in case of RNA, then the pentose sugar is ribose sugar. Okay, and in case of DNA, it becomes deoxyribo, because DNA is deoxyribonucleic acid, right? Here it is ribonucleic acid, because it has ribose sugar. This pentose sugar, that is ribose sugar. Next, it also has a phosphate group. So a phosphate group, sugar, pentose sugar, and then nitrogenous base together is a nucleotide. Okay, and this nitrogenous base is also further categorized as purines and pyrimidines. Okay, under purines you have adenine and guanine, and under pyrimidines you have cytosine, uracil and thymine. And this point becomes very important because thymine is present only in DNA and uracil is present only in RNA. Okay, so in case of RNA, you will be having adenine, guanine, cytosine and uracil. And in case of DNA, you will be having adenine, guanine, cytosine and thymine. Okay, so that difference you must have to know. Okay. Next is, what are the functions of RNA? There are certain viruses which are RNA viruses. Okay, in such cases, in those viruses, RNA will be carrying the genetic material. Generally, DNA is the one which carries the genetic material, right? But here in some viruses, RNA will be carrying genetic material because those are RNA viruses which will not be having DNA at all. For example, COVID virus. Okay, COVID virus is a RNA virus. Okay, it will not be having DNA. That is why for its test we go for RT-PCR. That is reverse transcriptase polymerase chain reaction. Okay, so that RNA has to be converted to DNA, then only we can analyze whether there is presence of the virus or not. Okay, so that is why we are going for RT-PCR, that is reverse transcription, that is we are converting RNA to DNA. This distinction you must know. Okay, other than that, what is the main job of RNA? Okay, this is a unique case because it is found only in some viruses. Okay, RNA viruses are very few in number. Okay, when we compare with other organisms, the population of other organisms. The main job of RNA is to transfer the genetic code. Here it carries the genetic material, but here it transfers. It does not have genetic material, but it transfers the genetic code needed for creation of protein from where to where? From nucleus to the ribosome. So how will it be? If there is a cell and inside the cell you have nucleus and inside nucleus you have DNA. Okay, and this DNA will be carrying genetic material and genetic code for production of particular protein. Okay, a body needs protein, any organisms need protein for survival. Okay, and there will also be another site called ribosome. Okay, ribosome is the place where protein is getting synthesized in our body. Okay, each and every cell will be having ribosome, and in those cells only protein is getting synthesized. But for protein synthesis, some information has to be sent, right? That so and so protein has to get synthesized. Okay, that information is there in DNA, but that information has to reach ribosome, then only this synthesis will happen, this creation of protein will happen. So who is transferring the information from DNA to ribosome? These RNAs are the one who are transferring those information. So DNA will be there, and from that RNA will be produced, and those RNAs are called as messenger RNAs. Okay, here it is double stranded, but RNA is single stranded. We all know that. Okay, basics. So mRNA will be produced, and this mRNA nucleus, ribosome, mRNA will be produced, and this mRNA will enter ribosome to produce a particular protein. Okay, we'll see what are the types of mRNA in a short while. Okay, next, ribosome, we know it is the factory of protein. And what is protein? Protein is nothing but chain of amino acids. Okay, amino acid polymer or polypeptide. Proteins are also called as polypeptides. Okay, so polymer of amino acid is protein, and this process prevents DNA from having to leave the nucleus. This is also an important point. Okay, RNA is the one which is carrying the genetic material, and it is transferred to, it is entering the ribosome for protein synthesis. And in this process, the DNA does not have to come out of the nucleus because it is sending RNA with the genetic code. So the DNA, the genetic material, is protected inside the nucleus. So DNA and genetic code is protected from the damage because RNA is the one who is doing the work. Next is, without RNA, protein synthesis can never happen in body. Okay, so all these points are very important. Next is, what are the shape and structure, different shapes and structure of RNA? They come in variety of different shapes, and they are single stranded molecule with shorter chain of nucleotides. Okay, We all know that DNA is double helical structure. Okay, double helical, double stranded structure. But then RNA is single stranded. But there is a particular type of RNA called tRNA. Okay, those tRNA are stranded. Okay, but then they are intra-strand double helix. Okay, they are intra-strand double helical, like this. Okay, if you see this picture, you will understand. They are actually, it is actually single strand only, but then since it is taking this shape, it will be like it is double strand. Actually, it is not. Okay. Next, we have mRNA, and this is ribosome RNA. Inside ribosome, there is one RNA called ribosome RNA. Now, we will see what are the different types of RNA and what are its important functions. These are the three types of RNA. The first thing is mRNA. This is mRNA. Sorry. Yeah, here it is. mRNA. mRNA carries codes from DNA to ribosome for protein synthesis. Already we saw this, right? There will be genetic material and that genetic code will be transferred to ribosome through this mRNA. So that role is done by mRNA. And then we have something called transfer RNA or tRNA. So like the name suggests, what does it do? It transports specific amino acids to ribosome for protein synthesis. There will be amino acids distributed in the cell. But then for protein synthesis, if there is a need for a particular amino acid, then these tRNAs, that is transfer RNAs, will be carrying those amino acids and it will arrange it. Okay, and that arrangement process is done by ribosomal RNA or rRNA. Okay, so ribosomal RNA or rRNA assembles the amino acid brought by tRNA in a specific order. That specific order's information is given by this mRNA. Okay, so all the three will be working in tandem. Then only we can produce protein. Okay, so from mRNA to make protein made up of RNA by the nucleolus. Okay, so all these points are important. There are three types of RNA. What are its unique functions? All this is important. A question based on this may be asked. Next is, these terms are also important. Okay, transcription, we already know what is transcription. That is production of, synthesis of RNA from the DNA is called as transcription. Making RNA copy of a portion of DNA is called as transcription. Next, we have something called translation. What is translation? We also saw this, but then the term is for RNA directing the correct order of amino acids in a polypeptide. That is called as translation. Okay, so where does translation happen? It will happen in the ribosome. Okay, that is important. Next is transcriptome. What is transcriptome? Is set of all messenger RNAs of the molecule is called as transcriptome. In one cell or in a population of cell, the set of all messenger RNAs is called as transcriptome. Likewise, we have yet another term called genome. Genome means the set of all DNA in a cell is called as genome. Okay. Similarly, the set of all RNA is called as transcriptome. So all these terms are very important. So see this, there is DNA. From that, transcription is DNA forming RNA, and reverse transcription is RNA forming DNA. Okay, this is done. Then we have translation, that is RNA to protein synthesis. The information is shared, right? That is called as translation. So all the terms become important. This point is also important, okay? Because what is transcriptome? What does it signify? That we have to understand. It reflects something. It reflects the genes that are present, that are being actively expressed at any given time. Okay, so what does transcriptome reflects? It reflects the genes that are being actively expressed at any given time, because mRNA is the one, transcriptome means group of all mRNA. mRNA carries message from the genetic material. Okay, so it has genetic code. So when we say transcriptome, then we are meaning every mRNA, every protein synthesis. Okay, so these genetic material which is getting translated into protein synthesis process. Okay, that is what is getting reflected from the term transcriptome. Okay, so such statements are also important. So yeah, this is yet another picture where you can very easily understand this. You have nucleus. Inside nucleus you have DNA. From DNA, RNA is formed. This process is called as transcription. Okay, which RNA is formed? mRNA is formed, messenger RNA. Messenger RNA carries information, and these green bodies are ribosomes. Okay, so they will be carrying, this mRNA will be carrying the genetic code, and then it will enter into the ribosome. And see here, these bodies, that is tRNA. Okay, tRNA will be carrying amino acids. These dots are amino acids. They will be collecting amino acids, transporting amino acids into the ribosome. Okay, and then they will be attached like this. Okay, this attachment sequence is done by rRNA, that is ribosomal RNA. Okay, and the information is given by mRNA. Transportation is done by tRNA. So all these points are important, and this is how the protein is formed. So this is how the protein is synthesized. These amino acid chain is formed, right? That is called as protein or polypeptide. Okay. So transcription, translation, difference you must have to know. We'll go to the next topic. Okay. We'll understand what is this mRNA vaccine. Okay. We have understood what is mRNA, what is function. Okay. But then we started with mRNA vaccine, Gemcovac. Okay. So what is mRNA vaccine? What does it do is, it will teach our cells how to make protein that triggers immune response. Generally, mRNA has genetic code, right? Likewise, this mRNA vaccine will also be having genetic code which will teach our cells, okay, every cells of a body. What will it teach? It will teach the process of making protein which will trigger the immune response. So it will create antibodies against the antigen which is coming inside in the form of mRNA. Okay, so when we actually get infected by the virus, the original virus, then the antibodies will be already there to destroy those antigens, that is virus. Okay, so this is how the process works, mRNA vaccine. So I hope you understood the concepts very clearly. The next topic for today is solar ultraviolet imaging. What is it? Solar ultraviolet imaging telescope. We have Aditya L1 mission, India's solar mission. Okay, and in that we are going to send this telescope as one of the payloads. Okay, there are many payloads, and of that one of the payload is this solar ultraviolet imaging telescope. Who has developed it? Pune's Inter-University Center for Astronomy and Astrophysics. They are the one who has developed this, and it has been delivered to ISRO, okay, Indian Space Research Organization, and it will be an important payload of Aditya L1 mission, which is scheduled to be launched in mid-August 2023. Okay, so let us understand what is this telescope, where is it placed, and other things. Okay, this telescope aims to study the sun's ultraviolet emissions and capture high resolution image of sun's atmosphere. Okay, in sun's atmosphere we have multiple layers, like we have we in Earth's atmosphere have multiple layers, right? Troposphere, stratosphere, mesosphere, etc. Likewise, in sun's atmosphere also there is multiple layers like corona, chromosphere, photosphere, radioactive layer, and comes the inner core. Okay, solar core. Okay, so it will be analyzing this corona. Other than that, it will analyze the ultraviolet emissions from the sunlight. Okay, so all these points are important. That is why it is solar ultraviolet imaging telescope, because it focuses particularly on the ultraviolet emissions, and it will operate in far and near ultraviolet regions. In which wavelength? 200 to 400 nanometer wavelength it will be operating. Other than that, it will observe the hotter and more dynamic regions of sun's atmosphere, such as the transition region and the corona. So okay, we saw, right? This is corona, the outermost layer is corona, and then we have other regions, chromosphere and photospheres. All these are being observed by, will be observed by this particular telescope. Next is, what is its significance? Because of high emission and radiation, sun's atmosphere has not been studied much. Okay, there are multiple countries who are sending multiple telescopes to observe sun's images, its atmosphere and other things, but then we in India are also taking one such effort. Okay, so that is why it is important. So it is one of the most difficult things to study the outside the environment of the sun. Okay, so that is why this telescope's use is very important. Next is, it will enable scientists to unravel the secret of sun and its impact on earth and other planets. That is also important. Okay, next is, it will also measure the UV radiation. Okay, it is named ultraviolet. Okay, solar ultraviolet. So it will measure the UV radiation which is hazardous for skin of human skin will get affected if UV radiation is exposed much. Okay, so it will be measuring all those things. Okay, next is, this telescope will monitor sun's activity and provide early warning potential of solar flares and coronal mass ejection. Okay, solar flares and coronal mass ejections are radiations. Okay, it will not happen all the time, but when it happens, it will cause more damage to Earth. What are all the problems that we will be facing? It will affect our satellites and our communication systems, and also our power grids will be affected. Other than that, we also have human health impact due to this coronal mass ejection or solar flares. So with the use of this telescope, it will send early warning to humans that so and so flare and so coronal mass ejection will happen in this time. So we can take preventive measures, okay, to avoid damage from such events. Next is, what is this Aditya L1 mission? Aditya, what is L1? That also we must have to understand. So Aditya L1 is the first dedicated solar mission of ISRO, Indian Space Research Organization, and in this mission we are going to put the satellite, I mean the spacecraft, into the hollow orbit, okay, in Sun Earth Lagrange point L1. So what is Lagrange point? We have to understand in detail, and it will fly approximately 1.5 million kilometers. Such distance based things are also important. Previously in UPSC prelims, such distance based things are also asked. Okay, so we have to give importance to such details also. So it will be placed 1.5 million kilometers away from Earth, and at which point? L1 point. There are 5 Lagrange points, and of those 5, it will be placed in L1 point. Next is, it will provide regular images and updates on sun's surface phenomenon, okay, and other space weather events. Next, there are 7 payloads of Aditya L1 mission, and of that, one of the payload is this suit, that is solar ultraviolet imaging telescope. This is what we have developed recently. These are the other payloads. You can have a look at it. All these are also important. Okay, if any other equipment has been produced and it has been installed in the L1 mission, then you must have to give importance to that technology also. Say for example, if there is spectrometer or if there is magnetometer, you must have to give importance to the background of what is a spectrometer or what is a magnetometer and other things. Okay, so this becomes important. Next is, what is this L1? That we'll understand. What is Lagrange point? Lagrange point 1. If there is 2 great masses, then they will be attracted by a gravitational force, like we and sun are getting attracted by a gravitational force. When this, and see for example, we have sun, we have earth, they both are attracted by a gravitational force. At certain points in the space, this gravitational force will balance the centrifugal force of the smaller body, that is the earth. Okay, those points are called as Lagrange points. I hope you understand this. They are specific points in the space where the gravitational force between the 2 large bodies like sun and earth will balance centrifugal force felt by the smaller body. That here the smaller body is earth. Okay, there will be centrifugal force experienced by earth, which will get balanced by the gravitational force at certain points. Those points are called as Lagrange points, and in Earth Sun system we have 5 Lagrange points, okay, named L1, L2, L3, L4 and L5. So it is named after this French mathematician. His name is Joseph Louis Lagrange, and these can be used by spacecrafts, which can be used, these L1, I mean these Lagrange points can be used by the spacecrafts for reducing the fuel consumption. That is one main advantage. Other than that, it will also offer uninterrupted view of the sun. Okay, say for example, if there is an orbit like this, if the spacecraft is going to orbit like this, then there are chances, wait, this is sun and this is earth, and if there is going to be an orbit like this, okay, when the spacecraft is at this position, then there are chances that earth's shadow will fall on the spacecraft, so that a view to the sun will be lost. Okay, instead if we are going to place them at the Lagrange points, these are the Lagrange points, I'll show you the picture, like L1, L2, L3, like this, L4 and L5. Okay, at these locations there won't be any shadow faced. Okay, so uninterrupted view of sun can be obtained so that we can analyze and study the sun in a better way. Okay, so we'll see the positions of the Lagrange points. Okay, we have 5 Lagrange points. This is sun and this is earth. You have L1, L2, L3, L4 and L5. Of these, 3 are classified as unstable and 2 are classified as stable. Okay, L1, L2 and L3 are unstable because they lie along the line connecting the 2 large masses. Okay, along the line connecting the 2 large masses. What are the 2 large masses here? Sun and the earth. Okay, they are along the line connecting the 2 large masses, L1, L2 and L3. Okay, and L4 and L5 are stable Lagrange points because they are at the apex of the 2 equilateral triangles that have the large mass at their vertices. So the large masses are at the vertices, and at the apex you have those points. The large masses are at the vertices in this triangle. Okay, and similarly in this triangle the large masses are at the vertices, and you have those points at the apex, L4 and L5. And then here this point is also important, L4 will lead the orbit of the earth and L5 will follow the orbit. Okay, so L4 will be leading the orbit of the earth. Okay, earth is going to orbit like this, right? So it will lead the orbit of the earth and this will follow the orbit of the earth. So all these points are important with respect to Lagrange points. You must know what is Aditya L1 mission, what are the payloads which is used in it, and what are the uses of this particular telescope. So all these points are important for your exams. So with this we have come to end of today's session. I hope you all found the session to be very useful and informative. Let us see in the next session. Thank you all.